creating a polysaccharide mucous membrane around the R strain, therefore making the R strain virulent in causing the pneumonia disease. That were grown on culture media containing radioactive sulfur, their protein became radioactive. Remember that their protein became radioactive. Bacteriophage, what it does is it goes and attaches itself to the bacteria and its genetic material then enters into the bacterial cells. Hello everyone, a warm welcome to today's session on chapter 6 of 2nd PUC, that is molecular basis of inheritance. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about how exactly the DNA gets packed inside the nucleus of the cell, and also we learnt about the search for genetic material, wherein we learnt about the experiment that was conducted by a scientist, Frederick Griffith, using the Streptococcus pneumonia bacteria. In today's session, let us study about the other experiments that were conducted by various other scientists in order to prove that DNA is the genetic material. So to begin with, let us start with studying about the biochemical characterization of transformation principle. So who was the first person to have proposed the transforming principle? It was Frederick Griffith, right? We had seen about that in the previous session. But there was no biochemical related evidence for Frederick's experiment, right? So that is why there was a loophole there wherein people were not able to accept it to that extent. And during that time, it was two important scientists who actually came into work wherein they took up some biochemical characterization methods in order to prove that DNA is the genetic material. So prior to the work of Oswald Avery, Colin MacLeod and McLean Carty, the genetic material was thought to be protein. So earlier people believed that it was protein that was the genetic material which was carried from one organism to another. That is before the work conducted by Oswald Avery, Colin MacLeod and one more scientist, McLean McCarthy. But after these three scientists conducted experiments, that is, they worked on the experiments that were already conducted by Frederick Griffith. That is, Frederick Griffith had done an experiment about transforming principle, right? So they worked to determine the biochemical nature of transforming principle in the Griffith's experiment. And what they did was they purified the biochemicals, that is the proteins, the DNA, the RNA, etc. from the heat-killed strains. So, if you don't know what this heat-killed strain is, please go back to session 2 of this chapter and have a look. So, you'll understand better. So, the heat-killed strain, that is Streptococcus pneumonia bacteria, those which were heat-killed, from that heat-killed bacteria, they isolated the proteins, the DNA or the RNA and all that, and they purified them. And that purified forms were the ones that could transform live R cells into S cells. So that is what they said. So biochemically, it was actually the proteins, the DNA and all that was able to carry the genetic information from the heat-killed S strain to the R strain, creating a polysaccharide mucous membrane around the R strain, therefore making the R strain virulent in causing the pneumonia disease. So therefore, they discovered that DNA alone from S bacteria caused R bacteria to become transformed. That's true, right? Because nowhere Griffith had mentioned that DNA was carried from S strain to R strain. But he had said that there was something which actually transformed the R strain. So keeping Griffith's experiment in base, these three scientists actually, they conducted biochemical studies and they found out that it was DNA that carried the genetic material which actually changed or which transformed the R strain, making it virulent, therefore causing the pneumonia infection. So this was the finding. And they also discovered that the protein digesting enzyme that is proteases, and the RNA digesting enzyme, that is RNases, it did not affect the transformation. So, these protein digesting enzymes, RNA digesting enzymes are all involved in the transcription, translation process and all that, but none of these could affect the transformation. So, the transforming substance was not a protein or a RNA, but the transforming substance was a DNA. So, therefore, in specific, DNA is the genetic material and it is not the RNA or the protein that carries the genetic information. But what exactly carries the information or the genes from one generation to other? It is the DNA. So, digestion with DNAs did not, it inhibited the transformation, suggesting that the DNA caused the transformation. And therefore, finally, those three scientists, they concluded that DNA is the hereditary material. So, that was the first time when they came to know that DNA is the hereditary material which is carried from one generation to the other. But not all biologists were still convinced about that. So, what did these three scientists did? They took the estrine bacteria, then that is the heat-killed estrine bacteria. From that heat-killed estrine bacteria, they isolated the proteins, the RNA and the DNA. They purified them and did biochemical studies on them. And later on, when they did biochemical studies, how did they do the biochemical studies? They treated them with the digesting enzymes. That is, for example, they treated the protein with the protein digesting enzyme proteases, and then they again made an experiment wherein they found out that proteases was not responsible for bringing any transformation. Therefore, proteins are not responsible for transformation, and they also digested the RNA by using a RNA digesting enzyme, that is RNases. Again, they found out that even digesting the RNA also caused the mice to die. So, that is when they found out that even RNA was also not the genetic material that is responsible for the transformation. Then what they did? Then they took the DNase enzyme, digested the DNA enzyme, and they found out that this was the DNA was the one that actually caused the transformation. Therefore, they proved that DNA is the hereditary material for any transformation occurring in a particular organism. But in spite of all these experiments, scientists were not. So, the next experiment that was conducted by two important scientists, that is Alfred Hershey and Martha Chase, in the year 1952 was the one that actually gave a solid proof saying that DNA was a genetic material. So far the biochemical studies was conducted on DNA, wherein the people started to believe that DNA is the genetic material, and later on it is the experiment of these two scientists which gave a unequivocal proof that DNA is the genetic material that actually transfers the information or transfers the characters from the parents to the offspring. And these two scientists, they mainly worked with viruses that infect the bacteria called as the bacteriophages. So, bacteriophages are one of the important viruses. So, don't think that the name bacteriophages means it is a bacteria, but it is actually a virus that actually infects a bacteria. So, the bacteriophage, what it does is it goes and attaches itself to the bacteria, and its genetic material then enters into the bacterial cells. Bacteriophage that is there, if you look at the bacteriophage, it will have a structure like legs in the form of pins like this. So, these legs of foot will go and cling on to the surface of the bacteria, and once they cling on to the surface of the bacteria, they will release the genetic material into the bacteria. So, how did they conduct the experiment? We shall see in the next slide. Once they enter into the bacterial cell treats the viral genetic material as if it's its own genetic material, and subsequently it will start to manufacture more viruses particles inside it. That is, the replication of the viruses will take place. So, therefore, what happened? A lot of viruses, the number of viruses inside the bacterial cell will increase, and Hershey and Chase, therefore, they worked to discover whether it was a protein or a DNA from the virus that entered the bacteria. So, what happened here? They took a bacteria, they infected the bacteria with the bacteriophages. These bacteriophages, what they will do is, they'll go and cling on to the surface of the bacteria. This is how they go and cling on. So this is the bacteriophage. Bacteriophage, which is a virus. So they'll go and cling on to the surface. Once they cling on to the surface, what they will do is they will pass the genetic material into the bacteria, and these bacteria will start to think that it is their own genetic material. It will accept the genetic material and it will replicate the virus genetic material, therefore creating a number of genetic material inside it, either the protein or the DNA. So that is how the viruses enter into the bacteria. So therefore, but they were not sure whether it was the protein or whether it was the DNA that entered into the bacteria. So what they did was, in order to prove that further, what they did was they grew some viruses on a medium. And that media, it contained radioactive phosphorus. So they took a petri dish. To that petri dish, they prepared a media containing radioactive phosphorus and added to the petri dish. In that petri dish, now they inoculated or they grew the bacteriophages. So once they grew that bacteriophages and some others on media that contained radioactive sulfur. So they took consider two petri dishes, one containing radioactive phosphorus and the other containing radioactive sulfur. So in both of these also they grew the bacteriophages, that is the viruses. So viruses that were growing in the culture plate which contained radioactive phosphorus, they contained radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not. That is true, right? Because the backbone of DNA is made up of phosphate or phosphorus. So therefore, because DNA contains phosphorus, those viruses grown on the radioactive phosphorus containing media, this one particular that I have shown here. So those viruses, they contained the radioactive DNA, but they did not contain the radioactive protein. Why? Because the DNA is the one that is actually made up of the phosphorus. So that is the reason. Similarly, viruses that were growing on the media that is containing radioactive sulfur, they contained radioactive protein but they did not contain radioactive DNA. So don't get confused here. Here the viruses contained radio DNA. Here the viruses contained protein. Why? Because DNA is made up of phosphorus. The backbone of DNA is made up of phosphorus. Therefore, growing them in a radioactive phosphorus media made the DNA radioactive. Similarly, here on the other side, those viruses that were grown on the media containing sulfur, their protein got radioactive. So that is how they started with the experiment. And the radioactive phages that were there, they were allowed to attach to E. coli. So here we can understand this very clearly here. So what did they do? They grew one set of bacteriophage. What are bacteriophages? They are nothing but viruses, right? Those viruses that infect the bacteria. So one had radioactive sulfur. The other one had radioactive phosphorus. Now those bacteriophages or viruses that were grown on culture media containing radioactive sulfur, their protein became radioactive. Remember that their protein became radioactive. And those bacteria which were grown on radioactive phosphorus media, their DNA became radioactive because already in their DNA, the backbone of the DNA is made up of phosphorus. So that is why. Now after that, what did they do was, they infected this. So these are separate experiments, okay. So what they did was, first they took the radioactive sulfur labeled bacteriophages, then they took the bacteria. So this was the E. coli bacteria. They took the E. coli bacteria and they inoculated this virus into the E. coli bacteria. Now these went and attached themselves, the bacteriophages. What bacteriophages? Those radioactive labeled bacteriophages, it went and attached itself onto the bacteria. On the other hand, again here, one more bacteria is there, that is viruses are there, no, which were phosphorus labeled. Those phosphorus labeled viruses were also allowed to infect to this E. coli bacteria. they also went and attached themselves onto the cell of the bacteria. Next, what happened we shall look into. So, radioactive phages were allowed to attach to E. coli bacteria. Then as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. So, the virus, it is covered by a protein coat, right? That capsid that is there, this particular thing, that diamond shaped thing, it is nothing but the capsid. So, that capsid is nothing but made up of proteins. So, that protein coat that is there should be removed. So, what they did was, they wanted to remove this portion, because once the infection progressed, what happened? The radio-labeled sulfur protein will enter into this particular bacteria. On this side, one more experiment, the radio-labeled phosphorus DNA will enter into this particular bacteria. Now, this portion is not needed. They should get removed. So, to remove that, what they did was, they agitated it, means they put it in a shaker. So, when it was shaking, what happened? These protein capsules that were there, or capsids that were there, they got separated from the bacterial cell. So, by blending experiment, they got separated because they were agitated in a blender. And the virus particles were separated from the bacteria by spinning them in a centrifuge, and they were put into a centrifuge. So, centrifuge is a small instrument which has the capacity, when you switch it on and adjust a particular rotation speed, it will start to rotate. Because of the speed of the rotation, what happens? Whatever unwanted particles that are there, which are heavier, that will get separated. Same thing happened here. The viruses got separated, that is, the bacteriophages got separated from the bacteria cell. Now, what is there here after centrifugation? Can you see here? Viruses completely got separated. What is there now? Only the bacteria. In this experiment, only the bacteria containing the radioactive sulfur, radioactive proteins, and in this, only the bacteria containing the phosphorus radioactive DNA is there. So much we came to know. So, next what they did was, the bacteria which was infected with the virus that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the viruses to the bacteria. So, they found out that later on, after removing everything, they went in for studying whether this particular bacteria is radioactive or this one is radioactive. And they concluded that those bacteria which had transferred the sulfur, right? So, this is a protein containing the sulfur. They were not radioactive. But those bacteria which transferred the DNA containing the phosphorus, they became radioactive. So, therefore, they understood that DNA is the genetic material that is transferred, but it is not the protein. So, that is when people started, or scientists all over the world came to a conclusion, and they started to believe the experiment that was conducted by Hershey Chase, though Griffith had put forward the idea. Actually, it was Mendel. From Mendel, it went to Griffith. From Griffith, further scientists, Avery, McLeod, McCarthy, all these scientists had conducted biochemical experiments. Later on, Hershey and Chase conducted this particular experiment, and they were able to prove that it is DNA that is the genetic material, or the hereditary material that is carried from one organism to the other. So, therefore, bacteria that were infected with viruses that had radioactive proteins were not radioactive. So, this you need to keep in mind. So, therefore, those bacteria which contained, so this one, right, because this bacteria's DNA was radio labeled. On the other hand, the other side, the bacteria's protein was sulfur radio labeled. So, therefore, because DNA got transferred here and DNA became radio labeled, there was radio, that is, these were the ones, they proved that DNA is the one that is actually, they had the radioactive proteins were not radioactive. That is, the bacteria that were infected with viruses that had radioactive proteins were not active, whereas bacteria that got infected with the DNA that was radio labeled, that was radioactive. active. So this indicates that proteins did not enter the bacteria from the viruses, but it was the DNA that was the genetic material that passed from viruses to the bacteria. So this was how they proved it. So this was about Hershey and Chase experiment. So from this particular experiment, we came to know that between the proteins and the DNA, it is the DNA that is the genetic material, and unequivocally all the scientists believed that DNA is the genetic material. So thanks to the experiments that was conducted by Hershey and Chase, and it became established as a fact that DNA is the one that acts as a genetic material. And however, subsequently it became clear that in some viruses, RNA can also be the genetic material. Say for example, it's not just DNA, but RNA can also act as a genetic material. Say for example, we have the tobacco mosaic virus, the QB bacteriophage. In all that, they have the RNA. So therefore, RNA acts as the genetic material. Those viruses that have the DNA can act as the genetic material. So this was concluded. So Hershey and Chase was the two scientists who actually with proper experiments was able to satisfy the question stating that DNA is the genetic material. So this was about this particular experiment. So now we shall study about the properties of the genetic material, that is DNA versus RNA. So genetic material, we learned that DNA can also be the genetic material, and in some viruses RNA can also be the genetic material. So a molecule that acts as a genetic material, it needs to fulfill certain criteria. What are those criteria we should see? So it should be able to generate replica, that is, it should be able to make copies of itself, that is, it should be able to replicate, that is, replication mechanism should occur in that particular genetic material. Then only we can consider that as a genetic material. And it should chemically and structurally be stable. They have to be quite stable. We subject it to some heat or some experimental chemicals or whatever. They should not get altered. Structurally and chemically, they have to be stable. Then only we can consider it as a genetic material. Therefore, both the DNA as well as RNA is also a genetic material, but DNA is more structurally stable than that of RNA. We shall look into it in the coming slides. And one more criteria that anything has to be a genetic material, it should provide the scope for slow changes, that is, very slow mutations should occur, because if mutations occur, if changes in the genes or present in the DNA occur, then only evolution can take place and change is needed, right? Otherwise, would we have evolved from monkeys? Not possible. So that is why very slow mutations at a very slower rate should be there in a genetic material, and it should be able to express itself in the form of Mendelian characters. That is, Mendelian characters, what all were there? That is, if there is a tall plant, if there is a short plant, either they cannot produce an intermediate character. Either the plant should be tall or it should be completely short or dwarf. So like that, it should be able to express and should satisfy whatever Mendel's principles were there. So all these are the properties for a genetic material. So since we know about that, now let us understand whether DNA, because we know the properties of a genetic material now, it will be easy for us to understand whether DNA is a good genetic material or whether RNA is a better genetic material. So we shall find out which is a better genetic material. So the genetic material should be stable. We had read, right? Chemically and structurally, the genetic material has to be stable. It should not get altered. It should not change. So the genetic material should be stable enough. It should be stable enough not to change with different stages of life cycle, or once that particular organism ages, it should not change, or it should not cause any changes in the physiology of the organism. So it should not alter the physiology of the organism, or once the organism starts to age, the genetic material should not get altered, and during the different stages of the life cycle of an organism also, the genetic material should not get altered. It should remain as such without changing. It should remain stable. And stability is one of the important factors or important properties of the genetic material, which is very, very evident in Griffith's transforming principle. That was true, right? In Griffith's transforming principle, the genetic material was stable because the S strain bacteria, though it was heat killed, what would we have thought? Okay, the bacteria died. The bacteria lost its virulence. It lost its capability to infect, cause pneumonia infection. But did the genetic material get altered? No, right? The genetic material passed on to the R strain, and in the R strain, it was supposed to make a polysaccharide mucous membrane, and it made it, right? It did not get altered. So that is when they said that. And what was the genetic material then? It was DNA, right? But they didn't know it was DNA. But it was DNA. So therefore, DNA is a better genetic material when compared to RNA because it is more stable in that sense. That is Griffith's transforming principle. I'll not read this again because I've already told it. And this now can easily be explained in light of the DNA that the two strands being complementary, if separated by heating, they come together when appropriate conditions are provided. So, this is also one of the stability. The structure of the DNA should not get altered. We conduct a lot of experiments, right? And during that experiments, we subject the DNA to a certain temperature. When that DNA is subjected to a certain temperature, what happens? Because of the breakage of the hydrogen bonds between them, the DNA that is wound like this, they will start to separate at one point. They might start to separate at one point. So once they separate and the experiment is complete or the transcription translation process is complete, they have to wind back, right? So that they have to be capable of winding back and getting back to their normal double helical nature. So that is about this. So therefore DNA is capable of doing so. So therefore DNA is a better genetic material. And further, the 2' OH group present at every nucleotide in the RNA. What is a nucleotide? Nucleotide is nothing but the RNA molecule having a ribose sugar unit and a nitrogenous base attached to it, right? That is a nucleotide. So in a nucleotide, the 2' OH, so say for example, if this is the 1', the 2', 3, then we have 4', 5'. So in the 2' OH group, so where in the 2' OH is present only in ribose sugar. A deoxyribose sugar in the 2', there is no extra OH group. So that is the difference we had learned in the previous session, right? You can have a look into those sessions there. So further, the 2' OH group that is present in the RNA nucleotide is a reactive group, and therefore because it is understood, because in the 2' region of the ribose sugar that is present in the RNA, because there in the 2' region OH hydroxyl group is present, it makes RNA more reactive to any of the structural changes. So therefore, it makes RNA quite labile and easily degradable. But DNA in the 2' region, they don't have a hydroxyl group. Therefore, the deoxyribose sugar is more stable than the ribose sugar. Therefore, DNA is more stable than the RNA. And RNA is also known to be catalytic. Therefore, it is highly reactive. But DNA is chemically less reactive and structurally it is more stable when compared to RNA. Why? One of the reason is DNA, no matter even though it is subjected to heat, after the heat is being removed and all that, after the temperature is normalized, the unwound strands will again start to wind back. That is one of the reason. And next reason is the deoxyribose sugar doesn't have a OH group in the 2' region, whereas the ribose sugar has a OH group in the 2' region. Therefore, the ribose sugar is less stable when compared to that of the DNA. So, therefore, now we have understood so far DNA is the better genetic material than the RNA. And therefore, among the two nucleic acids, the DNA is a better genetic material. And apart from that, in DNA, there is presence of thymine, right? And that is, in fact, the presence of thymine at the place of uracil also confers additional stability to DNA. So, RNA has uracil, right? So we know that DNA. Then I'll write it here, RNA. So DNA, what is it made up of? It is made up of adenine, that base pairs with thymine, then guanine with cytosine. But here adenine base pairs with uracil and guanine base pairs with cytosine. They do not have thymine. DNA has thymine, therefore it is more stable. RNA doesn't have thymine. Instead, they have uracil. That is why they are less stable than the DNA. And both RNA and DNA, both are capable of mutation. But how fast they mutate? Because I told you in the previous slide, if a genetic material has to be considered good, then the mutation rate should be slow. Both DNA and RNA will mutate, but the mutation rate changes. Say for example, RNA is more unstable because they mutate at a faster rate, which is not good. Whereas DNA, they mutate at a very slower rate. And that is why viruses having RNA as their genetic material, they have a shorter lifespan because they mutate and evolve faster. So that is why coronavirus and all, it's very difficult to find a proper medicine for viruses because of this particular reason. Because every now and then we would have found a medicine that can curb that particular virus, but very soon within a short span of time they would have mutated and they would have started to produce some other forms of proteins which these medicines cannot act on. So that is why it is very difficult to control those viruses, because some of the viruses, they have RNA as their genetic material and RNA mutates very fast. Therefore, any genetic material that mutates very fast cannot be considered a good genetic material. So that is why DNA, the mutation in DNA is very slow. Therefore, DNA can be considered a better genetic material than the RNA. And RNA can directly code for the synthesis of proteins. Hence, can easily express the characters. But DNA, can it directly code for proteins? No, right? Because in DNA, from DNA, mRNA molecule will be produced. That mRNA molecule will code for proteins. DNA will not directly code for proteins. So all that you will study in detail in the transcription translation section of this particular chapter. So DNA, however, is dependent on RNA for synthesis of proteins, and protein synthesizing machinery has evolved around RNA. This one point is good with that of RNA, but rest all the points was against RNA, thus making DNA a better genetic material. So this is about the properties of the genetic material, and also all these indicates that both RNA and DNA can function as genetic material. That's true, because they are capable of transforming information, they are capable of mutating, and they're capable of replication. All those things is fine, but because of certain factors or criteria which I had explained earlier, DNA is more stable and it is the most preferred genetic material for storing information when compared to that of the RNA. And for the transmission of genetic information, RNA is better. That's true, right? Because DNA will not directly transmit the genetic information in the form of proteins, because DNA takes the help of RNA to transmit the information. But RNA itself directly can transmit the information. But in spite of this point, rest all the point is against RNA. So therefore, DNA is more stable and it is best preferred genetic material when compared to that of RNA. So this was about this session. And in the coming session, we shall discuss about the RNA world. And also we shall discuss about the process replication. And replication is one of the important factors or important criteria for a good genetic material. What happens in replication, all that we shall see in the coming session. So I hope you understood this session. We shall meet again in the coming session. Thank you.